Welcome to the deep dive. We're about to embark on a highly intricate exploration of a microscopic world that is, honestly, quite literally shaping the planet.
Right.
And, actively shaping the biology of the person listening to this right now. Today, our source material is a remarkably detailed Wikipedia article covering this massive, incredibly diverse class of bacteria known as the Alpha Proteobacteria.
Yeah, the sheer scope of this bacterial class is, it's staggering. We are looking at a group of organisms that have infiltrated almost every conceivable ecological niche on Earth.
Wow.
They are in the soil, they are dominating the open oceans, and they are literally embedded in our own evolutionary history.
Okay, so our mission for this deep dive is to sort of demystify this complex bacterial family. We're going to make sense of the intense scientific debates surrounding how they evolved, trace their rather chaotic family tree.
Very chaotic.
Right. And uncover why they are absolutely crucial to human life, marine ecosystems, and the absolute cutting edge of modern genetic engineering. Okay, let's unpack this. We are looking at a class of bacteria within a massive phylum called Pseudomonadota.
Which is?
Oh, and if you happen to be reading older biology literature, you're likely going to see them referred to by their former name, Proteobacteria, or sometimes the Alpha Proteobacteria.
Exactly. And physically, the vast majority of these microbes are what biologists classify as gram-negative,
Right.
which is a crucial structural distinction.
Right.
When we say gram-negative, we are talking about the architecture of the bacterium's cell envelope. These bacteria have a thin structural layer sandwiched between two lipid membranes. That structural layer is made of a mesh-like polymer called peptidoglycan. It provides shape and rigidity.
Right out of the gate, though, the Alpha Proteobacteria throw us this huge biological curveball. Because while most of them fit that gram-negative profile perfectly, our source notes that some of the intracellular parasites in this group, they actually lack that structural peptidoglycan layer entirely.
Yeah, they just don't have it.
That missing armor makes them what scientists call gram-variable, which allows them to be incredibly stealthy when they're invading host cells. But before we get too deep into the cellular mechanics, we need to establish the stakes here.
The why should I care factor.
Exactly. Why should you, listening to this right now, dedicate your time to understanding this specific class of bacteria?
It's a totally fair question, because on the surface, this sounds like a super niche topic for microbiologists.
Yeah.
But the Alpha Proteobacteria family tree directly connects to the energy-producing factories operating inside your own body at this very moment. You have a deeply personal, ancient relationship with the organisms described in the source material.
We're definitely going to explore that evolutionary merger in a minute. But first, we need to map out the sheer extremes of this family. When we think of a biological family, we usually look for shared traits.
Or common survival strategies.
Right. But the Alpha Proteobacteria are defined by their contradictions. Despite sharing an ancient common ancestor, they have almost zero commonalities in how they live or what they consume.
It is a masterclass in adaptive radiation. They have evolved to exploit wildly different environments.
The examples provided in the text highlight this perfectly. On one end of the spectrum, you have the genus Methylobacterium.
The C1 eaters.
Yes. These are bacteria that metabolize C1 compounds. That means they feed on incredibly simple single-carbon molecules like methanol or formaldehyde.
Compounds that are highly toxic to most other forms of life.
Exactly. Then, completely on the other end of the lifestyle spectrum, you have Rhizobium. These are beneficial plant symbionts. They literally partner up with the root systems of legumes to fix nitrogen from the air.
Acting as a natural microscopic fertilizer that helps the plant grow.
And the contrasts don't stop there. You also have Wolbachia, which are endosymbionts of arthropods.
Right, they live directly inside the cells of insects. And they actually manipulate their host's reproduction to ensure their own survival.
Which is wild. And alongside them, you have the Rickettsia genus, which are aggressive, specialized intracellular pathogens responsible for severe human diseases. They share a lineage, yet their survival mechanisms could not be more different.
What's fascinating here is how this diversity translates into a massive global footprint. The source highlights one specific bacterium that completely recontextualizes the scale of this family.
Pelagibacter ubique.
That's the one. This is classified as an aerobic anoxygenic phototrophic bacterium.
Okay, let's break that down for the listener.
So it uses light to generate energy, just like a plant. But it doesn't produce oxygen as a byproduct. and it requires an oxygen-rich environment to survive.
But what makes it so significant isn't just his metabolism, right?
No, it's the sheer abundance. This single bacterium is so successfully adapted and so widely distributed across the globe that researchers estimate it might make up over 10% of the entire open ocean microbial community.
10% of the open ocean.
Yeah.
We are talking about billions upon billions of these organisms, just quietly acting as a foundational pillar of the marine ecosystem.
It's almost unfathomable.
But as massive as their presence is in the ocean, the most profound impact of the Alpha Proteobacteria is actually found inside us. This brings us to the eukaryotic elephant in the room, the Endosymbiotic Theory.
This is where it gets really personal for the listener.
The source explicitly notes that the Alpha Proteobacteria class is the sister to the Protomitochondrium.
Right. So if we trace biological history back roughly 1.5 billion years, we find a eukaryotic ancestor, a more complex, single-celled organism that had a distinct nucleus.
Okay.
This ancestor encountered a bacterium from this exact Alpha Proteobacterial lineage, but instead of a predatory event where the larger cell digested the smaller one...
Or a parasitic event where the bacterium killed the host.
Exactly. Instead of that, they formed a permanent functional partnership.
The eukaryotic ancestor swallowed the bacterium, and that engulfed microbe eventually lost its independence, evolving into the mitochondria.
And those mitochondria are the organelles responsible for generating ATP. That is the chemical energy that powers complex eukaryotic life.
Which includes every single cell in your body.
Every single one. When you move, when you breathe, when your neurons fire to process the information in this deep dive, the energy facilitating those actions is being generated by the highly evolved ancient descendants of the Alpha Proteobacteria living inside your cells.
It fundamentally changes how you view your own biology. We are talking ecosystems powered by a billion-year-old microbial merger.
Beautifully put.
But tracing the exact origins of that merger, figuring out exactly which branch of the Alpha Proteobacteria family tree our mitochondria sprouted from, that has caused major friction in the scientific community.
Oh, absolutely. The genomic data is incredibly messy.
Here's where it gets really interesting.
Yeah.
Because there are deep disagreements muddying the phylogenetic relationships.
It is a phylogenetic puzzle that has frustrated researchers for decades. A significant portion of this debate centers around 3 specific orders mentioned in the source: Pelagiobacteriales, Rickettsiales, and Holospirales.
Okay.
When geneticists sequence the DNA of species within these groups, they consistently find that they possess AT-rich genomes.
To visualize what that means, we have to look at the basic building blocks of DNA. The genetic code is written in base pairs. Adenine pairs with thymine, creating the AT pairs, and guanine pairs with cytosine, the GC pairs.
Right.
So an AT-rich genome simply means these specific bacteria have a significantly higher concentration of adenine and thymine base pairs compared to guanine and cytosine.
And in traditional phylogenetics, shared genetic quirks are usually a strong indicator of shared ancestry.
That makes sense.
If multiple organisms have highly specific AT-rich genomes, the logical assumption is that they belong on the same branch of the evolutionary tree. They must have inherited that trait from a recent common parent.
But this assumption has led to what researchers call artifactual clustering, which is essentially a massive data illusion. The AT-rich genomes aren't evidence of a shared parent. They are evidence of a shared survival strategy. This is a classic case of convergent evolution.
The theory is that these different bacterial orders faced similar extreme environmental pressures over millions of years. For Pelagibacter ubique, it was the nutrient-poor open ocean.
And for Rickettsiales, it was the highly restricted environment inside a host cell.
Exactly. To survive, they independently underwent genome streamlining. They aggressively shed excess genetic baggage to become as efficient as possible.
They optimized their genomes to require the absolute minimum amount of energy and resources to replicate.
And by pure biological coincidence, that streamlining process resulted in similar AT-rich genomes across entirely different lineages.
It is like looking at 2 completely unrelated hikers who independently decided to pack the exact same ultralight, specialized survival gear because they were heading into similar harsh climate.
That's a perfect analogy.
If you only looked at their backpacks, you might assume they planned the trip together, but they didn't. And the source adds another layer of complexity that drives geneticists crazy.
The ribosomal RNA issue?
Yes. The standard method for mapping out ancient prokaryotic family trees involves analyzing ribosomal RNA, the core machinery that translates genetic code into proteins. But for some of these bacteria, the GC content of their ribosomal RNA doesn't even match the GC content of their overall genome.
Which is a staggering contradiction. The source highlights the order Holospiralis to illustrate this problem. The ribosomal RNA of Holospiralis has a remarkably high GC content.
Wait.
However, when you analyze their complete genome, the overall data suggests they are more closely related to species with high genomic GC contents rather than to the other AT-rich orders like Pellagibacteriales and Rickettsiales.
It is like finding a highly complex engine manual where the chapters detailing the core machinery are written in a completely different dialect than the rest of the text.
It makes tracing the true evolutionary path incredibly difficult.
So this raises an important question. If the overarching ratios of AT and GC base pairs are creating data illusions, how do scientists actually solve this phylogenetic mystery?
The answer lies in highly specific molecular signatures, specifically something called indels.
Indels is a portmanteau for insertions and deletions. We are talking about conserved, rare structural changes found in widely distributed proteins.
You can think of an indel like a highly idiosyncratic, bizarre typo in a manuscript that gets copied over and over through the centuries.
Okay, I like this.
If you examine 10 different historical manuscripts found across the globe, and they all contain the exact same bizarre typo in the exact same sentence, you have definitive proof that they all originated from the same master copy.
Because the statistical probability of 10 different scribes making that identical complex mistake independently is practically zero.
Exactly. Using these conserved indels as molecular barcodes, researchers bypass the noise of the AT-rich genomes. They can accurately identify and assign new species to their proper place on the tree.
And the indel data has proven that the Alpha Proteobacteria actually branched off later than most other phyla and classes of bacteria, with the exception of the Beta Proteobacteria and Gamma Proteobacteria.
They are a relatively recent addition to the microbial world compared to older lineages. By following these reliable molecular markers, scientists have structured the Alpha Proteobacteria into 3 main subclasses: Magnetococcidae, Ricketsidae, and Collobacteridae.
But even with this structured approach, there is an outlier that refuses to fit neatly into the classification.
There's always an outlier.
Always. The basal group, Magnetococcidae. When we say basal in evolutionary biology, we are referring to the lineage that diverges earliest from the root of the tree.
Right. It sits at the very bottom, branching off before the rest of the family diversifies.
The source notes that this group contains a wide variety of magnetotactic bacteria, microbes that actually align themselves with the Earth's magnetic field. But only one single member has been formally described, Magnetococcus marinus.
And Magnetococcus marinus is at the center of a fierce taxonomic debate. There are strong arguments within the scientific community proposing that this basal group should be removed from the Alpha Proteobacteria entirely.
Completely kicked out of the family.
Some researchers advocate for elevating it to its own independent class, proposed as Candidatus Proteobacteria.
The exact placement of this basal group might seem like academic pedantry, but it is actually critical because of how it ties back to the story of our mitochondria.
Yes.
Recent phylogenomic studies suggest that the protomitochondrial lineage, the ancient ancestor of our cellular power plants, diverged very early on in this family's history. Specifically, the data points to a divergence occurring somewhere between the Magnetococcidae and the rest of the Alpha Proteobacterial taxa.
This early divergence dramatically rewrites our understanding of cellular evolution.
How so?
Well, for a long time, the prevailing hypothesis was that our mitochondria might be closely related to the Ricketsidae subclass. But if the protomitochondrial lineage split off near the basal root, it means our cellular ancestors established their symbiotic relationship long before the severe parasitic traits developed in the other branches of the family tree.
Which makes perfect evolutionary sense. A partnership built on mutual energy production likely wouldn't evolve from a lineage highly specialized in aggressive parasitism.
It would be a terrible foundation for a partnership.
And that distinction is vital, because we have to address the fact that while some Alpha Proteobacteria are powering our cells, others are actively trying to destroy them.
The dark side of the family.
The family contains some of the most specialized, ruthless pathogens in the bacterial kingdom.
The Ricketsiales order is particularly notorious. These are obligate intracellular pathogens. As we mentioned earlier, many of them lack structural components like the peptidoglycan layer.
Right, the gram variable ones.
Because they are structurally incomplete and have streamlined their genomes so aggressively, they are completely incapable of surviving on their own. They must invade a host's cells, hijack the internal machinery, and replicate until the cell is destroyed.
The real-world human impact of these pathogens has been historically devastating. The source highlights several specific culprits. You have Rickettsia prowazekii, the bacterium responsible for epidemic typhus.
Most brutal disease.
This is a disease spread by body lice that thrives in crowded, unsanitary conditions, and it has quite literally decimated armies and altered the course of human history.
Truly.
Then you have Rickettsia typhi, which causes murine typhus, typically transmitted by fleas, and perhaps most well-known in North America, Rickettsia rickettsii, the tick-borne pathogen behind Rocky Mountain spotted fever.
The stealth nature of these pathogens, their ability to bypass our primary defenses and hide directly inside our cells, makes them exceptionally difficult for the immune system to clear.
And the pathogenic members aren't limited to the Rickettsia genus.
No, the source also points to Brucella abortus, which causes brucellosis, a highly contagious zoonotic disease that jumps from livestock to humans.
There's also Bartonella henselae, the causative agent of cat scratch disease. So we have a family that encompasses the essential energy factories of life, the most abundant organisms in the ocean, and deadly intracellular parasites.
It's an incredible range.
But to fully understand the technological impact of the Alpha Proteobacteria, we have to look at the members of this family that operate as nature's genetic hackers.
This is where their biology intersects with modern human innovation. Some Alpha Proteobacteria have evolved the capacity for natural genetic transformation.
The source outlines this as a mechanism for genetic exchange, and the mechanics of it are wild. It is essentially an open-source code exchange out in the wild.
Unlike human reproduction, where genetic material is passed down vertically to offspring, natural genetic transformation is a horizontal process.
A bacterial cell will release fragments of its DNA into the surrounding medium, the soil or the water they're swimming in. Another competent bacterial cell will then actively absorb that loose genetic material from the environment.
It doesn't just absorb it, it physically integrates it.
How does that even work?
This happens through a process called homologous recombination. The recipient bacterium identifies sequences in the absorbed DNA that are similar to its own.
No.
And it systematically swaps out its old genetic code for the new fragments, permanently upgrading its own genome with traits gathered from its environment.
The source notes that this natural code swapping occurs in several of the soil-dwelling members we mentioned earlier, like Methylobacterium organophyllum and Bradyrhizobium japonicum.
But the undisputed superstar of this genetic hacking is a bacterium called Agrobacterium tumefaciens,
which is also referred to in the text as Rhizobium radiobacter.
Agrobacterium tumefaciens takes genetic transformation a step further. It doesn't just swap code with other bacteria. It possesses the remarkable ability to inject a specific segment of its own DNA directly into the genome of a plant.
In its natural environment, it does this to force the plant's cells to multiply rapidly, creating a structure called a crown gall tumor.
And this tumor then acts as a specialized factory, producing nutrients that feed the bacteria.
The bacterium literally overwrites the plant's genetic code to build itself a custom food source.
It's brilliant.
And this is where human ingenuity comes into play. Decades ago, scientists realized that if Agrobacterium tumefaciens was already acting as a microscopic delivery truck for genetic material, we could hijack the payload.
By removing the tumor-inducing genes from the bacterium's payload and replacing them with desirable genetic traits,
such as resistance to specific pests, tolerance to drought, or increased nutritional value.
Scientists created a highly efficient tool for genetic engineering. We allow the Agrobacterium to infect the target plant, and it dutifully delivers the custom DNA straight into the plant's genome exactly as it evolved to do.
It is the absolute foundation of modern agricultural biotechnology. The genetically modified crops that make up a massive portion of the global food supply are largely possible because we figured out how to co-opt the natural hacking capabilities of this single Alpha Proteobacterium.
It really is.
So what does this all mean? We have covered a vast microscopic landscape today. You have just explored a single, contradictory class of bacteria that manages to dominate the open oceans through sheer numbers, while simultaneously executing highly specialized, stealthy invasions of human cells to cause diseases like epidemic typhus.
We've navigated the chaotic genomic data of their evolution,
Mapped the early divergence of the basal groups.
and looked at how they naturally rewrite plant genetics in the soil.
And underpinning all of it is the realization that the ancient ancestors of these exact microbes are the biological engines keeping you alive right now.
If we connect this to the bigger picture, it reinforces the reality that biological diversity is never isolated. Ecosystems, both macro and micro, are defined by an ancient web of relationships.
We see predatory invasions that became permanent life-sustaining partnerships.
We see environmental pressures forcing totally different organisms to streamline their genomes in identical ways. Understanding these deep, invisible relationships is absolutely essential as we navigate a world increasingly driven by complex biological information.
It strips away the illusion that we are entirely separate from the microbial world around us. And on that note, I want to leave you with a final, provocative thought to mull over as you go about your day.
Always a good idea.
We know definitively that these bacteria undergo natural genetic transformation. They are out in the wild right now, sharing, absorbing, and integrating loose DNA into their own genomes through homologous recombination.
They are.
And we know that species like Agrobacterium tumefaciens naturally cross the barrier to overwrite plant genetics. Given all of that, what other microscopic cross-species genetic hacks are these shape-shifting bacteria silently performing on the ecosystems around you right now, completely undetected by our current science?
We are only just beginning to read the code.
We really are. Thank you for joining us on this deep dive into the hidden architecture of the microbial world. Keep questioning, keep exploring, and we will catch you on the next deep dive.